He's from Merck. Sir, are you present?
Yes, Dr. Mehta.
Sir, I'm just doing the PhD course. I'm not doctor. Yes. Talking about Mr. Arun Kumar Padmanabhan, he is joining us from the Merck team today. Just a second. Few glitches here. Sorry. So he has completed his post-graduation in drug design and biomedical science from Edinburgh Napier University. He has more than 13 years of industrial experience in several profiles from application scientist, production management, and marketing. In his current role as commercial marketing manager, he manages the protein research workflow products and immunoassay solutions portfolio in Merck. Sir will be talking to us about single cell technology today. Sir, you can take over.
Thank you, Mehta. I hope I'm audible and I'm sharing my screen. Could someone confirm if my screen is visible as well?
Yes, sir, your screen is visible.
Thank you. Very good afternoon to everyone. I would like to acknowledge and thank the IIT team for giving us this opportunity to present on our high sensitivity assay, which is called a single molecule counting. So before I start this, I'll give a very quick overview on why we need a very ultra high sensitive assays and the technology behind the single molecule counting, which we have developed, and what are the research areas where it can be applied. And then I'll finish my presentation with a short summary. To start off with, we all know that with immunoassays, what we are trying to measure is the biomarkers or any biological entity. And biological entity can be anything that can be that should be objectively measured, and it should also give us an indicator as a normal biological process or a pathogenic process, or that biological entity should also be an indicator of a pharmacological response to a therapeutic intervention. So this is one of the very well established definition of biomarker, which has been put together by the National Institutes of Health Biomarkers Definition Working Group. Not only this, the FDA has also classified the biomarkers as 7 different classifications based on the best glossary, which falls under the susceptibility, diagnostics, monitoring, prognostic, predictive, pharmacodynamic, response, and safety. So these are the 7 different classifications of biomarker. The reason why we need to measure the biomarker levels is that it will help us to understand 3 major questions. One, we could be able to differentiate a disease, or we could be able to identify a disease by studying a biomarker. And also it could also give us a differentiation between the normal sample and the disease sample. And if we talk about drug discovery, or even from a biopharmaceutical point of view, we can able to assess whether a particular drug which has been in development is actually toxic, or it is actually doing its internal purpose or not. And if you look at the classification of the biomarkers, right? So the current focus is on the preventive diagnostics. There's a well-known saying that prevention is better than cure. And if you look at the current health problems which occurs with us, it most of the 60% of the health problems occurs at around 30 to 45 years of age. And if we were able to identify those diseases at a very early stage, about 70% of the diseases can be averted. Only key thing is that we should be able to detect them at a very initial stages. So preventive diagnostic is a current focus, and we would need a high sensitivity assays just to look at those minor changes of the biomarkers to identify if there is a disease which is in progression or not. So that brings us the problem statement, right? So if we look at the current immunoassays, I'm comparing the ELISAs. If we look at the current immunoassay techniques, what we are able to measure is at the advanced disease state, because that's what the analytes are at the high level, and we would be able to measure them using this established technique. And if we could delineate the information which is available just above below that, then we would be able to identify 4 more levels of the diseases. We will be able to understand the healthy state, which is the endogenous level of the biomarkers, and a small increase in that, which could indicate an early disease stage, and then a moderate disease stage, and then advanced disease stage. So the focus of the single molecule counting, the technology that we have developed, is actually to measure the biomarker levels, which is below the detection limit. And what is the point in just detecting the biomarkers? We would also want to quantify them. The single molecule counting actually detects them, as well as it gives us the quantification of those biomarkers, so that we can come to a clinical endpoint informations. And apart from this, there is also two other challenges that we need to consider. Say, for example, if you want to establish a biomarker either as a diagnostic or even as a predictive biomarker, then we need to identify the right disease and the right population of patients to understand that particular biomarker. If we do not have that stratification of patients within a similar disease stage, then the information would be lost and we will not be able to validate a biomarker for that particular use. And the second challenge is if we are trying to establish a new biomarker, there is a bigger opportunity because if we look at the total human proteome, there are about 400,000 different proteins that is available. But unfortunately, we were able to measure only about 100,000 proteins because they are at the measurable levels. About 75% of the proteins are low abundance protein. So the current technologies of ELISA or a similar platform is not able to measure these proteins so that we are going to lose out a lot of information which is available. And this is one of the major reasons why there is only very few clinical markers comes to the marketplace every year. So if we have access to a technology which can give us a very high sensitivity information, then not only that, we'll be able to differentiate disease into multiple stages. We'll also be able to measure the endogenous level of an analyte to an elevated levels. We will be able to measure very small changes in the analyte level. We'll also be able to measure the biomarkers during the disease progression. If we can measure that small changes, then we can follow through the disease and we can study how the disease is progressing. And if it is a drug related discovery, drug development, we will be able to look at the pharmacokinetic profiles of a drug. When we can also do microdosing studies because of the high sensitivity assay that is available. We can identify new biomarkers and we can also discover new utility for an existing biomarker. Here's one example which I would like to present. So this is from a vaccine research center from National Institutes of Health, which was published last year, where they were trying to find a high sensitivity assay for an HIV antibodies. So they were doing a clinical trial where they are looking at the broadly neutralizing antibodies as one of the passive immunization treatment for HIV. And BRC101 is one of the candidate which they were studying because they could neutralize about 80% of the circulating human HIV strains. And they were trying to look at two clonal variants of the VRC01, which is VRC01LS and VRC07 523LS. And during the clinical trial, the samples that they were trying to assess is mostly the HIV port of entries, which are the mucosal membranes. Could be oral, could be rectal, or could be cervical. The challenge was the broadly neutralizing antibody levels were very low in these tissue types. And also the tissue itself is a challenging because it has a very complex matrix to it. So the team has tried to use the single molecule technology and they developed an assay which gave about 4 log of dynamic range when compared to the ECL standard method which is available at that time. Not only that, they were also able to bring down the lower limit of quantification to 120 picogram per mol, and they would able to follow through the antibody levels through two different passive immunization injections, on day 0 and on day 8. And they were able to measure the antibody levels in 3 different sample matrices: cervical, rectal, and oral. With the study, they were able to determine the clinical antibody threshold concentrations, which is required to mediate an HIV protection against HIV. Not only that, they were also able to determine the dosing regimes and also plan for a future clinical trials, given that they have access to a high sensitivity assays now. So this is one example. The other example is the Th17 pathway, which is one of the critical pathway within the immune system. And Th17 is also heavily related to the immune and the autoimmune disorders. And within Th17, IL-17 is one of the critical cytokine, which is a pro-inflammatory cytokine, which is also a target for multiple drugs for autoimmune disease, such as SLE. And the problem with the IL-17 measurement is there was no a standard technique or standard technology to give us the high sensitivity assays. And by using the single molecule counting, we were able to develop the very first high sensitivity assay for IL-17, not only just for the IL-17, for all the isoforms of IL-17, the IL-17A, IL-17F, and IL-17AF, starting from 4 femtogram per mol of concentration. That's the limit of detection that we have achieved using single molecule counting. So let me present you the core principle behind single molecule counting and why we have called it as a single molecule counting. The idea here is to identify those low abundance protein, which we are going to consider that as the golden needle among the bigger pile of proteins which is available. And the approach is to get sub-picogram quantification with minimal background and also a broad dynamic range. So the principle itself is based on the sandwich immunoassay, which is truly tested and tried core principle. So with sandwich immunoassay, what happens is we have the analytes of our interest, which is present in the sample, and then we use a capture antibody to capture that particular analyte, and then we use a secondary antibody, which could be either conjugated to dye or conjugated to an enzyme. And this will actually go and bind to the same analyte on a different epitope. In standard immunoassay, what we do is we stop the reaction here, and then we go for a reading at this particular point. With single molecule counting, what we have done is we expanded this particular step to 2 additional steps, where we use a proprietary elution buffer to break off this particular complex so that the analyte and the secondary antibody, which is conjugated to fluorescent dye, comes off into a free solution. And then this free solution is collected, and then we try to quantify the amount of fluorescence present in that particular sample. And this is achieved by using the system, the single molecule counting, SMC-X-Pro system. So these are the 2 proprietary steps which has been used in the single molecule counting. And within the platform, what happens is with the 96 cell plate, there is a 642 nanometer laser, which is a spiral laser. So it actually scans for the liquid within the liquid using a very focused integration point of about 276 microns. When this focused integration point scans the liquid, if there are any antibodies which is conjugated to dye comes into this integration point, then that particular dye gets excited and it releases a photon. These photons are captured by a low noise avalanche photodiode, and the data has been converted into a standard graph. And from that, we try to interpret the unknown samples. So this is the core principle of single molecule counting. So let me quickly present you a short video of the entire procedure and how the technique works. Allow me to reshare my screen with the volume.
Yes, sir, you can share.
Could you please could someone please confirm if my screen is visible?
Yes, it is.
And if the audio is coming through?
Yes, it is.
Okay, thank you.
Think of the possibilities. What would you do if you could detect the previously undetectable? The SMC-X-Pro is a fast and robust single-molecule counting technology with unprecedented sensitivity that allows the detection of low-abundance biomarkers in complex biological matrices. The technology captures and differentiates the true sample landscape to reveal data that was once lost in the background. With greater sensitivity, accuracy, precision, and reproducibility, SMC technology allows scientists to accelerate their drug discovery and development program by revealing previously undetectable, clinically relevant biomarkers. How does single molecule counting technology work? Let's take a closer look. Sample containing the analyte is captured onto beads coated with biomarker-specific antibody. Beads are washed to remove non-specific matrix proteins and a specific fluorescently conjugated detection antibody added. The antibody binds to the captured analyte to generate an immunocomplex. After washing to remove unbound proteins and non-specific material, the complex is disassociated. The beads are magnetically separated, and the final eluate is transferred into the final reading plate. Inside the SMC-X-Pro, single molecule counting takes place within a small interrogation space, illuminated by a laser within each well. The laser scans through the free-floating analyte suspension, fluorescently labeled detection antibody that passes through the interrogation space is excited by the laser and emits photons. These photons are detected by a low-noise avalanche photodiode and digitally counted as single molecule events. Interrogation of the sample within a small confocal window, coupled with the digital counting of individual fluorescently labeled detection antibodies, enables low background and a high peak signal, resulting in a broad dynamic range. The signal is analyzed with easy-to-use data analysis software that converts the data signal into response units, and the software algorithms compute the biomarker concentration. Our kits support various research areas.
So that is an overview of the single molecule counting. So let me give you a direct head-on-head comparison how the single molecule counting helps us in achieving a high sensitivity. So if we take a standard mouse IL-17 sandwich immunoassay, which has been done by the standard ELISA techniques, if I use the same antibody pair and if I try to translate that into a single molecule counting assay, either a plate-based or a bead-based method. So the technology allows me a flexibility to translate it into either of the plate or the bead-based assay. And if we could do that, what I get is I get a high sensitivity. I could move from the lower limit of quantification from 62.5 picogram from the ELISA down to 1 picogram or even down to 0.05 picogram if it is a bead-based SMC assay. Not only the improvement in the sensitivity, I could also move my sample volume requirement from 100 microliter down to less than 2 microliters. This is a very critical and very sensitive information when we are trying to study the clinical samples. And the technology as such is called single molecule counting. And the instrument that we have developed is called SMC X Pro. And this is the instrument that that has been developed by Merck. And this is actually a fluidics less instrument, which means that it comes with a very low maintenance and it can accommodate a 384 volt plate, which also means that we can run 496 volt plate assay simultaneously. It has a very rapid read time. Every single well takes about anywhere between 4 to 20 seconds of read time. And since there is no fluidics component involved into the design, it comes with a very low maintenance. And not only that, the instrument also comes with an assist program, which means it checks for about 18 different parameters even before we start the assay to make sure that the instrument is in good condition. We also provide an IQ, OQ, PQ validation services, and the instrument can be combined with any of the liquid handling automation available in the lab. And it gives a very broad dynamic range. Currently, it's about 4 to 5 logs. And it also comes with the integrated data analysis software. And we also support with 21 CFR Part 11 compliance if there is a requirement for biopharma or pharma industry. So as the video also said, the apart from the 2 application nodes that I presented, there are multiple other application nodes in which the single molecule counting has been used. And the research areas varies from inflammation, neuroscience, cardiovascular, immuno-oncology profiles to infectious diseases. And not only just the disease studies, we could also be, the system was also used in drug development processes to look at the PK/PD profiles and also the toxicity profile of the drugs. We have also developed an exclusive mobile app for SMC X Pro, and someone is interested in looking at the instrument in detail. The app can be downloaded by scanning the QR code, and the app comes with an augmented reality, which also projects the 3D model of the instrument onto the user space, and it can be very interactive. And all the information about the system, the technology behind the system, application notes, various research areas, the publication list, everything is pre-configured into the app, and that gives the complete information about the technology. Not just the instrument, we also support the SMC X Pro with the complete off-the-shelf kits. Currently, we have about 40 different validated off-the-shelf kits available. And we also provide the individual components to develop the complete SMC assay if there is a new target which we have to study. And we also provide a custom assay services, which means that the assay development and the sample testing can also be programmed with Merck. So with that, I would like to close my presentation, and I would like to thank the team again for giving us an opportunity to present the high sensitivity immunoassay system. Thank you. Happy to take any questions if there are any.
I'll request anyone from the listeners to give their any questions if they have. Characterize the binding of a small molecule DI. I would like to thank Mr. Anil Kumar. I will request all of the listeners to go to the main track for the remaining session.
Thank you, Medha.
Thank you, sir.
So the next presentation is by Dr. Ravi from the industry. Dr. Ravi has completed his PhD in biochemistry and structural biology from CSIR Institute of Microbial Technology, Chandigarh. His doctoral research was focused on the development of novel tag-based expression platform. His work served the base to design an affinity tag attached to any target proteins to form the protein complex. And before joining Sartorius, he has served as associate scientist at Lupin Technologies, Pune, and currently he is working as a field application scientist at Sartorius to look after the BLI-based label-free technologies. So I welcome Dr. Ravi for the next session. Sir, you are not audible.
Can you hear me?
Yeah, yes, we can hear you.
Thank you, ma'am, for the introduction. I shall share my screen for the presentation. Can you see my presentation?
Yes, sir, we can see the presentation. You can make it full screen and you can go ahead with the presentation.
Thank you. Thank you. So very good afternoon, everyone. So as ma'am has introduced me, I am Ravi. So I am working as a field application scientist with Sartorius for a specific technology, which is biolayer interferometry, and the brand name is Octet system. So Octet is a label-free technology which is used for the detection of biomolecular interactions basically. So here at proteomics facility, we have 8-channel instrument installed. We have different variants of the system on the basis of the throughput need. We have 2-channel, 4-channel, 8, 16 and 96 channels. Now this is a label-free technology and worked on the basis of dip and read method. That means we have a biosensor over which a ligand molecule is immobilized and that is dipped into your analyte solution. That means this system is completely fluidics free, which gives us an advantage to use crude sample matrix with the systems. Now using this system, what information you get? The information you get is the affinity, your rate constant. You can perform your cross reactivity assays with the instrument. You can completely replace your ELISA experiment. But in the field of drug development, you can screen out huge number of libraries in very small amount of time by performing an initial affinity based screening or yes no kind of screening. So I'll be showing you an example data for that in the coming slides. So first, what BLI can tell? As I mentioned, it is an instrument that is used to characterize the molecular interaction. So the first thing is it provides you the association and the dissociation rates of interaction. Also your kinetic affinity constants, your steady state affinity constants. You can also do quantification assays with the instrument directly from your crude samples, that is your serum samples, your supernatant, your lysates, your plasma samples, your mud samples, or whatever crude samples you have. You can also perform your cross reactivity assays with the instrument. Now, due to the technology and the absence of fluidics, it gives you a highly versatile it has a very highly versatile, what do you say, working so that you can measure a variety of molecular interaction with the system. You can screen out your small molecules or your aptamer libraries. You can perform your FC receptor binding or the reactivity assays. You can directly immobilize your virus samples over the biosensors and perform your assays. You can do glycan profiling. And for the industries, it can be the instrument is used for various QC or quality attributes determinations. So BLI has a, a role in every step of drug development, starting from your R&D, that is your identification of molecules, its characterization, and up to the final biomanufacturing and quality control attribute determination. So it can be used at each and every step of your development. So here is an example in which a carbonic hydrase protein used as an ligand molecule, which is immobilized over superstreptavidin biosensors with the use of avidin and biotin chemistry, and three different analytes with molecular weights of 330, 222, and 342 Daltons have been used to test their affinity. So you can see, and all of them are, all the experiments are repeated in triplicates. So this kind of data you will get from the Octet system for the small molecule analysis. The software also comprises with the fitting or analysis methodology. So it has non-linear curve fitting method and also your steady state analysis options available within the software for your kinetics determination and your steady state affinity determinations. So as I mentioned, it is a fluidics free so that you can use your crude lysate with the system. So you can see over here, two drug molecule, drug A and drug B has been screened for, different lysates has been screened with drug A and drug B molecules. That these are small molecules of molecular weight of around 300 Daltons. So with the crude lysate, you can see how clear is the data you will get, and you can rank them according to the affinities. Also, you can perform a yes/no kind of experiment for large libraries. So here is an example. You can see this is a positive data, that means your molecule is interacting, and this is a negative data, that is your molecule is not interacting at all. So likewise, you can screen a huge library in a very small amount of time and come up with your final screened out molecules to validate them with the titration series. So from hundreds of molecules, you can come down directly to 10 to 15 molecules, and from that, on the basis of affinities, you can screen out finally to 4 or 5 molecules. So it will save a huge amount of time and your investment also. This is an example in which at Michigan University, they have created derivative molecules, a library of derivative molecules, like, and they screen this library against a bromodomain protein, and these molecules have a molecular weight of around 133 to 200 Daltons. So you can see they finally get few positive results with the screening. So this is a publication in which they characterize the binding of a small molecule DI-591 with the DCN1 protein, and they find that this binding inhibits the interaction between the DCN1 and UBC12 protein, that in terms finally inhibits the nadylation of the UBC12 proteins. I can show you one of the example data which we have acquired from a pharma company at Syngene. I cannot disclose the name of the protein molecule and the drug molecule, but this is the kind of data, raw data, which you get from our instrument, and this is the analysis software. So you see, this is your baseline, your associations rates, and your dissociation data. After processing the data, that means subtracting your reference samples, normalizing your data, you'll finally get a fitted curves with your raw data to get all your parameters which has been required for your characterization. That means your affinity values, your rate constant, error in rate constant, and all other fitting parameters also, such as chi-square and your R-square values. So this is a solution for your library screening or fragment library screening that we provide. So I'll stop over here. So thank you so much, or for the organizing team for giving us the opportunity to present our technology. I guess tomorrow we have a larger session, so we can discuss and we can have an elaborate presentation to present this technology in a broader aspects. So thank you, ma'am. Thank you.
Thank you, sir. Do we have any questions from participants? If yes, you can ask now. Okay, so I think we don't have any questions, Dr. Ravi, for you. So, yeah, thank you for the presentation. We'll now close the session because this was the last presentation of this module. We can now move on to the general session which is going on. And then there we have few more sessions to attend before the lunch. So I'll now close the meeting from here and then we can go ahead with the common sessions. Thank you, Dr. Ravi, for the presentation.
Thank you so much. Thank you again.
It's not yet. Yeah, it's just visible, sir.
Yeah. That could be a relay delay. Yeah, that's right.
Yeah.
Great. Thanks for the organizers. Thanks to Professor Sanjeeva for giving me this opportunity. And good afternoon, team. Good afternoon, students. In my today's talk, I'll be talking about the Orbitrap mass spec with specific emphasis on the so-called structural mass spectrometry perspective. Right. So that is the core contents of today's session. In case if you guys have any sort of there is any kind of a relay delay or if there is any audibility issue, please feel free to put in the chat box. I can slow down accordingly. All right. To begin with, the contents of today's session I have plotted out here. Like I'll talk a little bit about the mass spec basics. Like I'm sure like you guys are much more familiar about this so-called mass spectrometer or the mass spectrometry. And then I'll talk a little bit about the trapping. Why are we talking about traps? I mean, the so-called orbit trap or ion trap. And how does this ion trap to orbit trap evolution take place? And a little bit about the working principle behind the orbit trap mass analyzer. So this would be a brief introduction. The most of the session I'll try to cover on the protein structure analysis perspective. Specifically, I'll try to emphasize on the cross-linking mass spectrometry. So that is one of the recently emerging techniques that has been routinely employed both in academic as well as in industry research. All right. To begin with, currently I have moved from slide 2 to slide 3. In slide 3, I'm trying to show you the schematics of any kind of a general mass spec. It could be LC or an MS, LCMS or a GCMS. In this schematics, if you could see, there are different components. I mean, the front end LC, I mean, the HPLC, capillary electrophoresis, GC, that is coupled to your mass spectrometer. Now, mass spec, it can be classified based on very many criteria. One criteria is the analyzer, choice of analyzer. That's what we call TOF, time of flight, the quadrupoles, ion traps or the orbit trap. The gist of today's content would be on the so-called high resolution orbit trap analyzer, right? So now let's go to the next slide. So now I have moved from slide 3 to slide 4. Where in this particular slide, I am trying to emphasize on trapping. I mean, why are traps, the so-called ion traps or orbit trap, important? So if you could see here, there is a statement that has been communicated by Professor Demmel. So he was the Nobel laureate who was awarded the Nobel Prize in Physics for one of his discoveries. So he says, a single trapped particle floating forever at rest in free space would be the ideal object for precision measurement. I mean, as a scientist or as a user, if you could trap a particle in a free space, and if you could trap for a longer amount of time, then that trapping phenomena itself will help you to precisely measure so on and so forth characteristics of that particular analyte or the particle. In this case, we are trying to measure the M by Z. That is how ion traps became very popular among mass spectrometers. All right. So now let's go to the next slide. So currently we have moved from slide 4 to slide 5. In slide 5, I am trying to show you the a table in which there is head-to-head comparison between different trapping instrument. I mean, the 2-dimensional ion traps and the 3-dimensional ion traps. The logic is that the trapping efficiency, trapping capacity has been improved. That's the take-home message. Like if you could see this particular slide, slide 5, the trapping efficiency has been improved approximately around 14% from 2D to 3D. So that is what has been achieved. So now that once you have increased the trapping efficiency, I mean, the amount of information that can be collected, sustained, the next thing is obviously, can we resolve the information? That is where orbit trap came into picture. So you trap more.
So the assisted hair study, but they have taken basic heart parameters. Authors
I think there is a background. Can I request the participants to be on mute?
So there are even those cardiac parameters, physiological parameters. Actually, cardiac basic, but their inference is based on cardiology.
Can I request the organizer to put others on mute?
I think I just saw the profile. One of them is a cardiac physiologist. That they have not mentioned in the first page. I personally don't think we need at this stage, but we can send it to reviewers.
Okay. For the benefit of clarity, clear, audible. So I have muted all the participants. In case if there are anything, you feel free to unmute yourself and you can talk. All right. So I was trying to migrate between ion trap to orbit trap. As I was telling you in the earlier slide, in the 2D to 3D ion trap evolution, one major thing that has been improvised is the trapping capacity. So once that you are able to trap more, the next thing would be obviously to resolve the trapped information. That is where orbit trap came into picture. So in this particular slide, slide 6, the central cylindrical structure that you see is the orbit trap mass analyzer, which actually detects analytes based on the image current, employing FT, Fourier transform, as the algorithm. So that is what is being shown in this particular slide. This slide gives you the overall working principle in a nutshell. I mean, if you could see here, there are 3 color rings. The 3 color rings corresponds to 3 different ion population. They move around the central electrode on a fixed path. This fixed path is called orbit. That is why from a scientific standpoint, we call it orbital trapping of ions, orbit trap. This movement causes some sort of a image current. It generates something of image current. So that image current is what is being translated or transformed using FT, and ultimately it is being read as the M by Z spectral plot. This is the overall working principle in a nutshell. Now let's go to the next slide. Slide 7, I'm trying to show you the different protein structure analysis tool. I'm sure most of you must be familiar with some of the tools that has been shown. It could be X-ray diffraction, it could be NMR, tri-electron microscopy, ion mobility, HDX, and so on and so forth. For today's session, we will be talking about this XLMS, the one that is being shown on the right-hand side bottom panel, which is nothing but cross-linking mass spectrometry. Right. So what does mass spec do in the name of XLMS? Right. That is what I will be showing you in the next slides. So currently we have moved to slide 9, which gives you an overview about the different structural workflows, structural methods that can be performed using a mass spectrometer. If you could see on the left-hand side, it could be native MS or cross-linking MS. On the right-hand side, I have plotted about top-down, hydrogen deuterium exchange, and of course the protein digestion-based schemes. So these are different technical approaches through which one can delineate, one can retrieve some of the limited structural information of a given protein backbone using mass spec as a technique of choice. All right. So now let's go to the next slide. So in recent times, if you could see, mass spectrometer in combination with be it NMR or tri-electron microscopy, it has been routinely used in the name so-called integrative structural biology approach, wherein they are trying to solve the structure of very complex protein, or it could be even the virion protein complex. All right. So now let's go to the next slide. So how does it work? I mean, the so-called XLM, I mean, cross-linking mass spec or the structural mass spec, how does it work? So we are in slide 12. In slide 12, I am trying to show you a general schematic wherein you are subjecting a sample to enzymatic digestion and then you are injecting into the mass spec. So the mass spec generates something like this one. This is the spectral data, MSMS spectral data that has been generated. So using this data, you tend to identify the sequence. So this is the general mass spec scheme. Now let us go from a general scheme to a cross-linking scheme. We have now moved to slide 13, wherein I am trying to show what does a cross-linking spectrum looks like. So in the earlier slide, you could have seen a simple plot which would deliver a sequence, a peptide amino acid sequence. Whereas in this particular slide, I am trying to show you, in addition to amino acid sequence, we also have some signature fragments. So the signature fragments along with the cross-link. So this is how mass spec can be used to identify the cross-linked peptide amino acid regions between two proteins, or it could be within a given protein. So if you could see in this particular slide, there are red color ones which might correspond to one protein population, and the blue ones which might correspond to another protein population. I mean fragments from two different populations. And there are also purple or violet ones which corresponds to the cross-linked peptide population. So these are the signature patterns that will be identified, which in turn will be helpful to identify the cross-linked peptide regions between two proteins or within a given protein molecule. All right. So now we have moved to slide 14. This particular slide is more about the method. I mean, in case if you guys have a chance, you can visit Professor Sanjeeva's lab, wherein he has a tribrid instrument. In that particular instrument, you can do this kind of a method. So either you can do an MSMS method, a CAD MSMS method, or a CAD MS followed by ETD MSMS method. So these are different technical approaches which is routinely being used by most of the laboratories to decipher the cross-linked peptide population. We are now moving to slide 15. So in slide 15, I am trying to show you a spectral plot. How does the CAD only data looks like? And on the that is on the left-hand side panel. And on the right-hand side, I am trying to show you CAD followed by ETD, dual fragmentation. So the benefit is that in CAD, you get one sort of information wherein you get the cross-linked peptide population. And on ETD, we will be able to confirm. It is not only the precursor or the cross-linked fragment, it is also the fragment that is originating from the cross-linked fragment. So you get one more layer of information, one more layer of confidence. So these are different ways of technically validating your cross-linked peptide population. All right, now we have moved to slide 16, which again shows another technical approach, a technical method which can be employed. In this case, it is CAD followed by targeted mass triggered followed by HCD. The logic is that we are instructing the instrument. If you are finding one particular fragment, one particular signature ion, then you can go for one more round of fragmentation. You are instructing the instrument to trigger one more round of fragmentation, provided so on and so forth of signature pattern is being observed. So this is also another approach, complementary approach that has been employed to identify a cross-linked peptide. So, and if you could see in this, the next slide, slide 17. So this slide shows you the overview of the spectral plot of CAD, MS2 followed by one more round, MS3. So it is MS2, MS3 fragmentation. I'll quickly go through the animation wherein how we are doing this particular fragmentation event. Like we are targeting selected signatures, selected MYZs that is observed in a fragment, and those fragments will be fragmented once more. That's why it's called MS3. So by using this MS2 followed by MS3 approach, we are able to accurately identify, yes, indeed, these two fragments from two different peptide populations or cross-linked. So that is what has been shown in this particular slide, slide 17. All right. Finally, we'll now quickly navigate through slide 18. So all the subsequent slides, like 18, 19, 20, it is all about the overall workflow using this software. The name of the software is Proteome Discoverer, and the node that we use is called Xlinks. So this particular node, it has been developed in collaboration with Utrecht University, Netherlands. So this particular node is being used to identify the cross-linked peptide population in a given sample, whether it is purified protein or whether it could be a complex sample. So slide 18 followed by slide 19, it shows the software data analysis workflow. How this can be defined, I mean how the cross-link can be defined and how it can be identified. So we are trying to automate, semi-automate the overall data analysis workflow. And this is how it has been done. So now we'll move to slide 20. In case if you slide 20 is visible. So here I'm just trying to show one of the annotated spectral plot, wherein you will be able to see peptide A and peptide B. So this is how a cross-linked peptide, two different peptide looks like. So cross-linked peptide on the top panel and cross-linked fragment on the bottom panel. Fine. So this is how this particular workflow is being done. Finally, to conclude, the choice of liquid chromatography mass spec approach matters, specifically when you're trying to do any kind of protein-protein or cross-linking interaction studies. And above that, MS resolution at the MSMS level. So what kind of resolution is possible in this particular hardware? At the fragment level, that is important because you are trying to differentiate, discriminate cross-linked species from normal canonical species. Canonical in the sense, the one that is being routinely observed as a part of the tryptic peptide. The next important thing is experimental design. So the choice of cross-linkers, are we using MS cleavable, MS non-cleavable? All these technical things matters a lot. The third important thing is computational tools. The choice of computational tools is equally important. Above all, though this particular workflow looks a bit automated, what I would say is manual curation or intervention is a must, because that is how we will be able to filter out biologically relevant, technically significant information from the one that is not relevant. So with this, I would like to conclude. I'd like to thank again the organizers and Professor Sanjeeva for giving me this opportunity. Thank you, and if there are any queries, I'll be happy to answer.
Is there any question from the audience? You can ask here, or we'll share the email ID of Dr. Saravanan. So you can just write to him and he'll get back to you with the questions. So
we will meet in the evening session.
yeah, so we would post the we will post your mail ID, sir, in our chat box. So you might get questions from our audience. So you can address them in the convenience at your convenience. Okay. So anything
Sure,
that
sure. That should be fine.
yeah. So thank you so much to everyone in this track. So we will end this track right now and we have a short lunch break. After that, I think you guys have received the different track links for our evening.
Now we have industry talk from Citivia, and we will welcome Dr. Vipin Kumar. Dr. Vipin Kumar has completed doctorate from Department of Bioscience and Bioengineering of IIT Bombay. Has 7 years of experience in the field of proteomics technique. His expertise includes gel-based proteomics, protein purification using the ACTA system, protein analysis using BioCore or Western blotting techniques, NGS, etc. His focus is to manage unpredictable requirements for the specific skill sets and provide product and application training to user. Currently, he is associated with Cetiva as an application scientist, where he leads the proteomics range of product across the South Asia region. We'll welcome Dr. Vipin Kumar.
Yeah, thank you, Avasthi, for our introduction. Let me share this screen and then I'll start. Hope the screen is visible and I am also audible.
Yes.
Yeah,
Yes,
thank you
you're
for
visible.
confirmation. Yeah, thank you for confirmation. So hello everyone. So as I seen that this track is for focusing mainly for the nanomedicine. But what I'm going to cover in a next 15 minutes here, like you may have used the gel based proteomics, you may have run the SGS page, Western blot, right? And then 2-dimensional. So how those conventional techniques are very useful in industry. So that I'm going to talk about how those conventional techniques are helping in a production of biopharmaceutical product, then how you can apply those techniques, those learning which you are learning right now in industries. So that I'm going to talk about in my next 10-15 minutes. So there is a one thing, there is a host cell protein. Okay, so whenever we are going to produce any biopharmaceutical product or any biologics, right? So when we express the specific protein in a cell, so along with my target protein, there are other proteins also get expressed, which is a host cell proteins. Okay, so along with pharmaceutical products, there will be a host cell proteins will be there, which will be considered as a impurities. So when you are going to bring this product into the market, when you are going to inject into the patient, right, or the in animal, so you have to remove those host cell proteins, which will be considered as impurities. If you will not remove those host cell protein impurities from the your pharmaceutical product, right? So it may affect the efficacy of your molecules, means this pharmaceutical product. It may, sometimes it may be a toxic for your patient because there are lots of proteins which can be a toxic for the patients or for the animals, right? And then with time, they may generate some immunogenicity. So that's why it's very important. You have to remove the host cell proteins from your pharmaceutical product. And this will be considered as a host cell protein as impurities. So how is the process for the production of the monoclonal antibody purification or be any biopharmaceutical product? So you do the cell culture, right? You produce some specific biologics, and then along with there is a impurities also there. Then you start the purification. So when you start the purification, so you do a different step of the purification, and then you ensure with steps those impurities will get removed. So if you see here, there's a lots of impurities are there. Then you done next step of the protein purification, then next step, and then in the last there will be some impurities will be there. You cannot remove 100%, but there will be some amount will be there. So before putting this product into the market, you have to measure how much impurities are there, and these impurities are host cell proteins. So that has to be measured, and each step you have to see like how much impurities you are removing so that you can optimize your protein purification experiments. Okay, so how we measure now it comes. So there is a guidelines are there, regulatory guidelines there which says ki ELISA is a standard method, and they have put the guidelines. Okay, you have to do ELISA and then you have to measure. So how the ELISA works for those? So when you do the purification, right, along with your proteins, there's a host cell proteins are there. These are the host cell proteins are there. So what we have in ELISA plates, we having a pool of antibodies against those host cell proteins, which is already coated onto the plate. This will be considered as a primary antibody. Then you put your pharmaceutical products, which you have purified. If there is a host cell proteins will be there, it will go and then bind there. And then you put a secondary antibody, which again, it's the antibodies against the host cell proteins. Now, if impurities are there, if host cell proteins are there, it will detect and then give a color. So on the basis you can measure, you can find out what is the concentration of the host cell proteins are there. So that measurement can be done by using a ELISA kit. Now, one limitation comes. The limitation is because here we having a pool of antibodies. So we have to ensure, do we having a sufficient amount of antibodies, or do we having a all type of antibodies also to detect that protein, to detect that HCPs. So that's why then you have to do a coverage assay. So coverage assay says, okay, we having 100 proteins, and do we having all 100 antibodies to detect or not? So that we have to find out. Do we having all antibodies to cover those all proteins or not? So that we have to find out and then say key how much cover, how much we are covering those protein, and that is the coverage assay which you have to do in industry. So in industry, people do ELISA to quantify. Then they have to validate that ELISA kit. This is the kit which is correct or not. Then they have to do a coverage assay. So during the coverage essay, then they have to do a complete 2D experiment, which shows like how you can do it. So I'll go how the process is for that. So you may have done SGSPs, or some of you have done the 2D also. So now if you're having a 3 samples, you have to run 3 different gels are there, or maybe you have done the 2D dyes also, where a dye is involved. In this case, there is an advance, there is a dive, 2D dive. Dive then says differential in blot electrophoresis. So what is the steps if you see here? Now in this case, you having a samples which can say host cell protein. So you can say cell lysate, which is labeled with Cy3. So these are the fluorescence dye. Okay, so you have done the labeling and then you have done the first dimension. So you have done the separation of your proteins on the basis of isoelectric point on IPG strip. Once this separation is done, then you take the same IPG strip and then put on the second dimension. Okay, SGSPs, where you are going to separate the protein on the basis of molecular weight. And then you transfer those proteins on a membrane. So here if you see, this is the membrane which presenting all proteins, all host cell proteins. Okay, now what you do, you having a antibodies, the secondary antibodies or the primary antibodies, which you have generated against those proteins. But when you generate the antibodies, it is not sure you are like generating 100%, right? There will be some proteins which are not immunogenic. So you may not able to achieve 100%. But if it is not 100%, then how much? It is 60%, 70%, 80%. That has to be documented. And that's why we do the coverage assay. So in this case, this membrane is presenting total proteins. Then we having antibodies which we have generated, and then it's labeled with Cy5 dye. Okay, and then we do the immunodetection. So now this is the blot which we have, and then we do the basically image capture, the scan at a Typhoon, and then we get a one image. Now, if you scan at Cy3, we get the presentation of all proteins. If you scan at Cy5 wavelength, then we get a antibody presentation. So here, if you can say, like if there are total 10 proteins are there in this case, and then here we are able to detect only 5, then we can say there are 50% detection is there. Means we are able to generate only 50% of antibodies, or we can say the coverage is 50%. So this is the steps how people do the coverage assays to validate the ELISA kit in industry. This is how the data looks. This is Cy3 gel, which is presenting to your total proteins. This is Cy5, where the antibodies presentation are there, which basically against the SCT. And this is the overlay, and then you get the total common proteins, how much you are able to cover protein. So that is how you're getting around 93% coverage. So this is how the data looks for any like ELISA, HCP ELISA, and then the coverage. So how the overview, if you see the what things are required. So if you see in any proteomics lab, all those things will be available. So the moral what I want to convey, so whatever the technique is there, learn it properly, because they having their very, very good application in the industry. So to measure the whole cell proteins in a pharmaceutical product, what you need quantification and then validation of that ELISA. So for the quantification, then always ELISA kit is there, which quantify, which measure the whole cell proteins. Once you have measured the quantification, once you have know this is the amount of the whole cell proteins along with your product, then you have to validate that kit this kit is correct or not. And this is the workflow then again. So initially you do the sample preparations where there are different kits are involved, like quant kit, cleanup kit, and then illustrator. So these are the different kits which will be helpful for your sample preparations. Now you will be having a host cell lysate, or you will be having antibodies which you have generated, right? So you can label those using a Cy3, Cy5. Once labeling is done, then it comes to a protein separation, where you do the first dimension of the IPG for machine and then 2D. So they are like complete 2D setup, where you are going to use IPG strip first dimension, and then 2D second dimension, separation on the molecular weight. So all those instruments will be there. Once this is done, then it comes to a simple protein transfer, which you may have done during the Western blot, right? So you will be having a membrane, then transfer unit, which you can transfer all proteins from the gel onto the membrane, right? Then again, it comes to a simple Western blotting, where you are detecting your proteins using a generated antibodies. Okay, once now you having a plot which is developed, then you do the image acquisition, which using a Typhoon scanner or IQ800 instrument, which already there in the central facility. Now it comes to a once you having a blot, then you have to do an image analysis, because one gel having around 500 or some 1,000, 2,000 number of these spots, right? So you cannot do the analysis using a naked eye, right? So there is a software is required which do the complete analysis of your image and then tell how much proteins are there in the primary image and then how many spots are there in the secondary image, and then it will tell you the common proteins. So this is how you can see the like complete workflow of HCP and to validate those ELISA kit. Yeah. Thank you so much. If you will be having any question that we can go through and then we can discuss. So I kept around 5 minutes for the discussion. Do let me know if you have questions.
Yeah, thank you very much for this talk. So I'll now request to have some questions from the audience.
I know it's very less time to present a complete work, because just I want to give a feel. These are the techniques are very, very important when you are doing in a master's or during the PhD, initial time of PhD, right? The SGS space, Western blot. So I think it's a very simple, but yeah, they're having a very good impact in future applications, like especially 2D, I'll say. So if you learn now 2D, it has a big advantage in industry. And we will be having a like complete setup, like for the 2D electrophoresis, from sample preparation to till complete image analysis.
So just one question I had is because of this various gel to gel variabilities it is comes. So what are the levels of skills or expertise one should have to minimize those? Can it be done through data analysis, or one has to be very much skilled?
Yeah, I think this is a very great question. So generally, like what happens when you run, like if you having a two gel, if you having a two samples and then you are running a two different gel, so simple the gel to gel variation will come. You cannot compare, right? When you run the two different gel, the electrophoresis issue will be there. But that's why this is the advanced technique. This is a dive. So here you are running a one samples and it detecting the same gel. So here you are comparing within the gel. So this is the one gel, right? Only a single gel. Then transfer, and then you are detecting on the same blot. And then from the one blot, you are getting a two different images like this one and then this one. So here the gel to gel variation will be reduced. That is the one. Now, when you will be having a three samples, so you can run a one standard samples, which is common in across the all experiment. So there is another die which is a Site2. So you can take a one pool sample and that pool sample is labeled with Site2. Okay. And then whenever you run experiment 1, 2, 3, 4, so run that pool also. Now using that pool samples, gel to gel variation can be normalized. So during the data analysis, then you can normalize the data using a Site2 die or the Site2 sample. So that can be done.
Okay. So thank you for giving this insight. Uh, so I just request any other question if anyone has.
So this is the just like how we can see. This is the like one gel, and we're getting a three images. So there's no gel to gel variation. And then we can completely normalize the data using a Site2. Yeah. So if you have question, we can discuss or.
Hello, sir. I would like to ask a question.
Yeah.
Could you tell the relevance of ELISA and 2D? How these two techniques are like
Yeah.
if we can compare and
Right. So there is
that.
no. So there is no comparison. Basically, both are the like complementary technique. ELISA is to quantify the host cell protein. Okay, and then 2D is to validate that ELISA kit. Ki are we doing a correct quantification or not? Are we having all antibodies to quantify my protein or not? So ELISA is for quantification, and then 2D is to validate my ELISA kit, to validate the presence of all antibodies. Do we have all antibodies which can quantifying the protein, or do we having 80%, or do we having a 60% or 70%? So 2D is just to validate the ELISA kit. Hope you are
Thank
getting
you.
the
Thank you.
hope.
Yeah, I got the answer. This is more relevant to the people who are interested in joining the industry. So,
Yeah,
any
industry. Yeah.
advice? What of advice you want to give them? There, how, what, like, what should they focus more on if they are willing to join
Right.
industry?
Yeah. So what I suggest right now, actually, as if you are in a initial time of your PhD or the master, what I suggest that learn the technique whichever you are learning, just don't skip, because like what I have seen, people say, okay, no, there is a mass spec is there, then they leave the 2D technique. Okay, now 2D is no 2D is an obsolete technique. No, it's not. So learn as much as technique you can learn, because they having a good advantage in the industry in future. So during my MSc and then PhD, I done the lots of 2D experiment, and now I'm utilizing those techniques in industry now. And these the guidelines which came now in 2017 only. So all people has to do a ELISA also, and then they have to do a 2D also. And there is a lack of expertise because people have not done. Okay, so it's good to learn as much as technique you can learn during your coursework, during your PhD, during your masters. That I'll suggest, because they having advantage in future.
Thank you so much, Dr.
Yeah.
Vipin Kumar. Sorry. Thank you so much. Uh, Now I'll request Ekta, Dr. Ekta to take forward this meeting. Now all the participants will be joining in this link only.
Hello.
Yeah.
Am I audible, Nisha?
Yes, yes, you are.
So next up we have a session by Dr. Bikramjeet Basu. and he's going to talk about the Society for Biomaterials and Artificial Organs India. So I'll be sharing my screen so that we can have a little interview, a little introduction on him. Do we have Dr. Grunji Basu on, like, sir, have you joined? Hello. Lisha, am I audible?
Yes, you are. Yes, you are audible.
Okay, can you, has Professor Basu joined? Actually, I had
I could not see in the participants list.
Yeah, so we can wait for a while. He told me that he is connecting, so.
What I know about digital health. I saw some pretty high-tech stuff when I was here for the last few minutes, but this is a different kind of tech and digital health that I'll be talking about. As Amit said, I'm one of the co-founders of RIA. We're an insurtech company focused on driving better health outcomes for people with health insurance. And today I'm talking a little bit about India's digital health stack and how we're using it to improve health insurance and health outcomes. So I understand that most students here are already familiar with Ayushman Bharat's digital health mission, thanks to the close ties between KCDH, IIT Bombay and ABDM. But I'll just spend maybe a quick minute telling folks who don't know much, just giving a quick introduction. So ABDM is obviously defining some of the core building blocks for digital health in India. And we, as part of the industry and startup ecosystem, we're very excited because this has the potential to transform healthcare, much like UPI did for payments in India. So while you see many different building blocks here, they're sort of divided into three major layers, which are the user applications, the health interface layer, and the data exchange layer. And there are many different components, all of which are really important and add significant value to the ecosystem. But what I'm going to do is talk about one piece here and how we are leveraging that, which is the health information exchange. And one important part of the health information exchange is actually health data standardization. So why is health data standardization important? So if we look at the human body today, right, there's no doubt that it's very complex. There are thousands and thousands of conditions we can suffer from. There are thousands of procedures that can be done on us. There are tens of thousands of different medications out there that we can take, and many more, right? And from a terminology perspective, if we look at an example of the coronavirus, right, it's been referred to in so many different ways. There's Corona, COVID, COVID-19, SARS-CoV, then we have the variants which continue to grow. If we look at diabetes, and this is a search from a medical catalog called ICD, and I'll tell you a little bit about ICD shortly, but we see diabetes itself has so many different nuances and variations which are very specific, right, as you can see on the screen. If we, our health records, which last many, many years in time, we want to make sure that something diagnosed early on is easily understood, recognized later on in life by a different doctor, right? That same doctor may or may not be around. We might be in a different country, city. We don't want any kind of confusion in what our health record states, especially when we move to another doctor, provider, etc. Right? That's one thing I don't think I want anyone to get confused about. So that's why these systems for classifying, identifying, and communicating health data emerged in order to make sure that data is consistent, standardized, and understanding is also consistent. But of course, health data standardization is a very complex problem, right? if I look at just a few examples I put up on screen here, health data exists in various shapes, forms, and sizes, right? So here on screen on the left, you may see handwritten notes or prescriptions. You may see a printed format of a report. In the middle, you see different languages used, sometimes on the same form. You know, you see the Dolo written in English, but there's something written below in Hindi. And then, of course, you know, we have scans and images and X-rays of varying degrees. And you know, I know Amit has done significant work in pathology. I've not even put that up here, right? So we know that it's extremely diverse and complex. And how do we standardize this? So one part of how to standardize this brings us to medical coding. Today, if we look at medical coding globally, there are three major standards of medical coding out there. The first is called ICD, which is the International Classification of Diseases. The second one is called LOINC, which is used for lab observations primarily. And the third is SNOMED, which is used for clinical terms and symptoms, but is one of the most extensive of the three medical coding systems out there. All of these in some combination or the other are used across the world. Some countries are still evolving them. So if we look at ABDM and India's health stack, it has proposed the use of all three of these standards, as you see on the right-hand side. But also there are a few others mentioned here in terms of DICOM for diagnostic images and scans. And more importantly, one called FHIR, which is used for interoperability of health data. So any information of exchange of health data between systems, say a hospital and an insurer, your device, you know, your Fitbit, your Apple Watch, your Goki, and a certain other system. If it's not doing so today, it will be likely following these standards, including FHIR, for interoperability fairly soon. Companies like Apple and Google Health as well, in terms of their health part of the ecosystem, do follow some of these data standards. So how do we look to standardize this data if it's so complex? So one of the things we do is we take health data, which varies in all kinds of forms, shapes, volume, and velocity, and we bring it into a data lake in its raw form. Now, that includes things like medical scans, images, reports, activity feeds, so on and so forth, right? Now, when we have that raw data, then we start to run other tech on top of it to first digitize it. And again, I know this as a problem, some of you, especially Professor Amit and others in the ecosystem, have worked on previously, using image processing techniques and algorithms to extract data from images, convert it into text. But for us, the additional layer comes in trying to codify it using these medical data standards. And then, of course, apply medical rules and validation to make sure that the coding that we have obtained from these algorithms actually makes sense. And that's an important step. And as it continues to evolve and more data is available to us, AI also starts to come into the picture to help solve this problem. Now, once we've standardized some of this data, right, what I just described to you was that we took some data from various sources like labs, customer history, hospitals, so on. We applied some medical coding standards. We brought it into a standard form in our health data platform. There was additional customer data we have around customers, and then using that standard health record, we pass it into a core insurance and health platform, which we use for various use cases like underwriting, distribution, claims, and improving health. So let me take one use case and tell you a little bit more about what health insurance underwriting is and how do we use that standard health record there. So for those of you not familiar with health insurance underwriting. It's basically the process by which usually a medically trained person or often a medical doctor will take your health insurance application with all your health records that you might have submitted to them. They look at it, they evaluate it, they're trying to gauge your health risk, and based on that decide whether or not they want to give you an insurance policy or what kind of pricing you should get. Now, this is a complex procedure. It takes time. Sometimes human underwriters are sitting and processing hundreds of applications manually every day. In some cases, it may take 7 days to get a decision back on your application, right? So once we standardize the health data and we understand what are some of the conditions in your past. So if you look at this on screen, you see a kind of a rule set here. It says if you've had this condition represented by this code in the past, and there is some status, duration that we know that you had it for or had it in the past, this rule set will make a decision to say, yes, let's accept this risk, or let's, we need to conduct further tests, or it needs to be rejected for certain reason or the other. So rather than having human underwriters look at all this data manually, we're able to start to simplify, make the process quicker and faster through these kind of rules for now. And there are about 4 lakh medical rules that we've created around this. So you can imagine the complexity when you may have multiple conditions and so on and so forth. And then gradually, as we get more and more data, we build more intelligence and on top of that. So this is one example of how standardized health records can help us improve health insurance. Another example is in health claims. So one of the biggest complaints that people have about health insurance is that when you go into a hospital, when you're in a time of need, that's when some of these insurers don't come to your help, right? They take hours and hours, they make you sit there, do paperwork, then they might say, no, this is rejected for XYZ reason, or they may not even give you a reason. And then that's really not a time when you want to sit and wait and have to deal with all of this. So in a similar manner to what I described, when claims data, which is a lot of it is health data, comes into the system, you can digitize it, you can start to classify it, code it up using the health data standards, and then pass it through an engine where you can map it with a person's insurance policy. So for example, in this case, I mentioned if a person came in with a severe pneumonia and pneumonia has a ICD code of J13, you can look up the insurance policy and very quickly make a determination saying, hey, yes, this is okay to approve, and go ahead and instantaneously pass the claim and send details back to the provider. Right? So trying to make this easier and faster for a customer and improve the process of insurance. One final example that I do have is how do we use this standardized health data for improving health outcomes. So as more and more data comes in, right, think about your activity trackers and your medical records from labs and statements. As more and more data comes in, there are these clinical pathways and algorithms and all kinds of intelligence that researchers and organizations and startups around the world are building on top of some health data, which they're using to predict or to try to drive interventions in advance on people. Right? So having this standardized health data, which is fairly universal because it's using the same coding standards, allows us and others to use these kind of models, use these clinical pathways. The one you see here is actually one we have pulled from an international organization, and allows us to then drive intervention in healthcare. So for example, if someone shows an increase in weight, blood pressure, you can perhaps this pathway may suggest an adjustment in the diet or a change in medication, thereby obviously trying to make a customer healthier and healthier. So To summarize, of course, I wanted to just introduce everyone to a small part of what Ayushman Bharat Digital Mission's data standardization recommendations are and why they're important and how we are trying to use it as a startup in the space to drive improvements in health insurance and health outcomes for people as well. There's plenty of more. This is just scratching the surface, but would love to answer any questions if anyone might have. Thanks again to IIT Bombay and KCDH for having me.
Thanks a lot, Nipun. Any questions from the audience? So I have a question. So Nipun, this entire data standardization, format standardization exercise, that's quite a huge and has many stakeholders. So what are some of the most, or which are some of the most challenging stakeholders, and how do you think they can also be brought in under, they can also learn about the benefits of data standardization, and how do we drive that change?
One of the most challenging ones that we see today is actually the EHR and EMR space, because India, in terms of health record systems, even in hospitals, and I'm only considering, say, the easier ones of the lot, right? The hospitals which do have some kind of systems out there, the space is very fragmented. There are hundreds of different systems being used, and so there's barely any standardization. Investing in some of the tech to start to standardize is also expensive for them, right? So where there is motivation to use a system, even there it's often expensive, and we'll have to see how the government, even though it's recommending all of this, it is going to be a little bit of a gradual evolution to get there. Now, obviously, beyond that, I know that Ayushman Bharat, through its own health program and mission, is trying to take some of the data at the grassroots level that it does have and try to act on it. I don't know too much about the quality of data. I have heard various things from people, so that I won't get into that side of things, but focusing on the the part where there is some kind of digitization initiatives already happening, that I think is one of the more complex.
Okay. And are there like any compelling economic advantages for digitization to, let's say, small pharmacies or small clinics, if not in the short term, in the medium term, that can be used to convince them?
It's a great question. I think would have to think about that a bit more. But I mean, I think since you brought about pharmacies, one of the things that the government is trying to also do is to create a standardized drug registry as part of this digitization. And so by having, say, a standardized drug registry where you can start to easily look at prescriptions and figure out what what drugs need to be given without confusion, I think that part could lead to less errors. However, at the same time, we do know we're in an ecosystem where there's a lot of things happening off the books, and there's a lot of fraud, waste, and abuse in the system. Now, some of that, I'm afraid, unfortunately, does end up helping the small pharmacies and so on. So it's an interesting question, and we'll have to see how we can drive better economics in this. I don't have a good answer to that one.
Thanks. Professor Ganesh, you go ahead with your question.
Thanks. Yeah, your answer. Thanks for a nice talk, Mr. Nipun. Your answer actually led me to the next question. So given SNOMED and other kinds of standard efforts towards standardization, what is the scope for building in Indian languages? I mean, should it be translation? Should it be transliteration? Have there been standards that you've been part of there? And what is your experience overall dealing with multilinguality?
Sorry, can I I'm just wondering, when you're talking about the standards, is your question that should we look at an Indian standard, or is your question around, say, the problem
dealing
of
with multilinguality, like the point of standardization and drug names, right? But then with diseases, with ailments, with many other common nouns, maybe not even proper nouns, right? You'll need Indian language equivalents.
Right.
I mean, I know that there is some effort from the ABDM and others to have a so SNOMED localized, right? Localization.
Right.
So, what has been your on-ground experience with the with Indian language incorporation for common nouns, for verbs, etc.?
Yeah, actually, I was in a conversation about a couple of weeks ago with a much larger ecosystem provider, a platform in the Indian ecosystem. And they've actually started looking at some of the language-based customization on top of the existing ICDs and SNOMEDs. So rather than trying to build a new system, they're also looking at, okay, what are the various commonly used, and you have to start with the 80-20, right? So you pick the most commonly occurring conditions and so on, and you start to try to create mappings of those. So I think that's where some of the industry, and we've not reached that point, but that's what we're seeing some of the industry is starting to do. And there definitely is a need, but there, what they have is that, they still have doctors that work for them. So it's a more controlled environment, right? So they're able to look at, again, narrow the problem even more and more and say that, for the common cold, what are the various terms in, let's start with a localized geographical area, then we sort of expand it out. I think that's how they're going, but, I'd be fascinated to see if, some of the things happening in terms of localization globally in terms of technology, and I'm sure you and other people in IIT are leading a lot of interesting research and work out there to see if one's able to kind of even map medical terms like that. It would be very, very fascinating to see.
Thanks for that. So, would you be actually interested in collaborating with KCTH on the angle of multimodal input, audio input, and also languages, multiple languages? Is it something interesting? Because we have many other faculty members who.
Yeah, let's take that offline. He's very enthusiastic about collaboration, but let's not put him on the spot here.
Sure. Thanks.
I'd be happy to take that offline. Thanks.
Nice talk. Hearing you.
Any last questions? Okay, great. Thanks a lot, Nipun.
Right, thank you.
It gives me great pleasure to introduce our next speaker, Mr. Shashank Chilamkurti. He is an alumnus of IIT Bombay, and he has had quite an illustrious career so far, and hopefully going forward as well. He's a founding member and AI scientist at Cure.ai, which is one of the most successful medical imaging startups in the world. Perhaps it's not even no longer a startup. It's now it has FDA approvals and so forth. He's also a co-author of PyTorch AI framework, and his work involves deep learning on medical images obtained from radiology. And his passion lies at the intersection of AI, healthcare, and open source technology. He has contributed to the development of AI in healthcare and is instrumental in development of QER, which is a neuro AI tool. Over to you, Shashank.
Thanks for the interesting, Professor Sethi. I'm really blessed to be here, I think. Thanks for inviting me, KCTH. Being part of the, alumni. I've been part of the electrical engineering as well, which Professor Sethi works in, right? So I'm really blessed to be here. To be honest, a lot of stuff I learned from IIT Bombay, I literally apply them, like radon transform, posterior spares, which a lot of convex optimization courses that I have been part of. Professor Ganesh actually taught me that course, who just asked me a question. Who just asked a question, right, to the previous speaker. I'm really blessed to be here. So what I'm gonna do, let me, guys, can you guys see my screen?
Yes.
Okay, so what I'm going to do is I'm going to give you a pitch slash presentation of one of our, more specific products. So this is, for example, a page, a presentation I have given in a neurology department. So you can understand, so basically the idea is that I'm go really deep. Instead of giving you like upar ka funda, instead of giving you just about basic stuff, I wanted to dig deep and explain you like more neuroscience work that we ended up having to do, right? So I'll do that because as part of, so if you're a healthcare startup, you will realize very quickly that devil is in the details. You will quickly realize that's where most fun is as well, right? So that's what I will go deep into. Instead of giving you more philosophical or abstract things, I'll go really deep, right? So before that I'll just intro what we do, right? At Cure.ai, basically what we do is we apply artificial intelligence deep learning on medical images, basically X-rays, CT scans, ultrasounds, right? So right now, we have just raised, by the way, a 40 million round last week or a week before. So fairly well funded. We have about 100 plus employees, 150 employees today. Interestingly, we have, unlike other places, other AI startups in the space, we have very narrow focus on disease areas. Because again, devil is in the details, and each disease and each condition is different from other conditions. And we are a AI guys, data scientist dream. We have largest repository of 1 million images. 1 billion, sorry. I think I called it 1 billion. It's actually a 1 billion images that we have internally that we use to train our models. And every year we basically process 200 million images, right? So this is the overall different AIs that we have. We have an AI on CT of brain, we have AI on CT of lung, and we have an AI of X-ray of chest. So I'm going to specifically talk about, basically brain related stuff, neuroscience, right? And then we also have, we realize that it's also important for us to not just have AI, but to have a complete end-to-end solution. I'll talk about this as well in more detail. But overall, so this is a kind of footprint that we have today. We are in about 50 countries now. We have operations there, and we have about 400, I think it's 500 now, 500 plus sites across the world that are using our AI. So we impact about 10,000 patients every day. And I think, we are actually probably instrumental in at least actually impacting lives. Sorry, actually impacting critical, maybe 10,000 people, right? I can probably confidently say I have saved them using the algorithms that we built, right? And this is different people that we work with, a lot of ministries of health, a lot of universities, like Indian health, a lot of Indian places as well, Niti Aayog, all these people we work with, Gates Melinda Foundation, right? And we are very research heavy. We publish a lot, quite a bit. So we have 37 plus journal peer reviewed publications. If you count conference publications, I think it will be like 150 something. I think this month alone we submitted 6. Like March alone we submitted 6 conference proceedings. So we are very, very active in terms of, I think that 4 patents is now 6 US patents. So super active in terms of science. So collaborate quite a bit with a lot of people around the world. I think one of the best papers we have is the Lancet one. I don't know if you guys remember if you guys know about the Lancet, it's the most prestigious medicine journal in the world, right? For example, all the vaccine trials, right? They get published in the Lancet, for example. It's that impactful. So we have got our AI stuff published there. So this is in general about Cure. I will talk more about stroke. I will talk specifically stroke. Maybe I'll give you a brief overview of what stroke is and how the stroke treatment works. The stroke is essentially part of your brain not getting blood. Let's keep it very straightforward. So heart attack, if you know, is basically your heart muscles itself not getting blood, right? That's what heart attack is. And therefore your heart is dying and your heart has stopped working. Similarly, it's brain stroke, also called as brain attack, is when brain is not getting blood, and because brain is such an important part of the body, if the brain doesn't get blood, the person will die very quickly, right? It's as I mean, so if you don't treat the stroke patient within maybe a day or two, and you kept them without any care, they will die. I mean, patients will die. It's really life-threatening condition, right? And thankfully, there are treatments, and more treatments are coming. More treatments are already proven in this space. I'll talk more about the exact treatments, but overall, so the how the stroke patient is treated is also important to understand this discussion where I'm going to. So by the way, why I am going through this is because this is how our data scientists and our engineers and everybody in Kure thinks. We think we have to think extremely intense into actual clinical area, and then we also have to be good with data science and stuff. This is basically the bread and butter of us. So that's the reason why I'm giving you a taste of how working in AI startup in healthcare looks like, right? So I'm just going into one deep dive, doing a deep dive to give you the taste. So how the treatment look like is patient comes to the stroke center, right? Through the ambulance. Is there a question? I think I hear some echo. Is there a question? No. Okay. Good.
No, I think someone accidentally unmuted themselves. I will mute them.
Got it. No issues. So how it works is I'm just being quick on my time. Yeah. How it works is basically a stroke patient comes to the stroke center. And then what happens is basically they will be evaluated whether it's stroke or not. Then they will be immediately be taken to a CT scanner, where they will be taken a CT scan. So CT scan, computed tomography, right? And then radiologist will be looking at this CT scan, and he will give his opinion to the emergency doctor who looks at the, who basically ordered the CT, who was in charge of the patient. So emergency doctor confirms that yes, this is a stroke, and yes, this is this kind of stroke. Then he calls a neurologist, and he calls a neurologist. Neurologist is basically a doctor who is specialized in brain related stuff. Neurologist again looks for this, looks after all the data that is generated so far, whatever analysis of emergency doctor, radiology, CT scan, and the patient symptoms. He looks at all this information and he makes it. So he then decides, okay, what treatment do I give to this patient? So there are two kinds of treatments to the treatments that can be given to the a stroke patient. Number one is medical treatment, which is there is this specific drug called as TPA, right, or altiplase. So this particular what it does is this dissolves the clot, and then there is another set of treatment called as thrombectomy, which is more surgical. It's minimally invasive surgery. So surgeon will basically insert a catheter into the vessels, artery, and then will pull the clot out, right? So you don't need to know exact details, but know that there are two kinds of treatments. And neurologist basically makes a decision. Both of them are not lightly taken. So you'll have it's not like just give aspirin kind of thing. It's a lot more complicated than that. So neurologist basically decides based on all the information, and then he calls up surgeon who actually does the surgery in case it is required, right? So as you can see, this is, I think, briefly informative of pretty much like any kind of treatment that you get. It involves like many people. Like if you do any complicated, if you go with any complicated disease such as stroke, this is the kind of process that is followed. It involves multiple people. It involves like multiple data crunching. Let's say that whether it is done by, it's mostly done by humans at this point of time. And then all this takes time. It's not synchronized. So they have to call each other. Like for example, the information, all these arrows that you see, these are literally phone calls, like calling, hey, CT guy, can you please prioritize this patient? This patient is a stroke guy. He requires immediate CT scan. And then the CT scan guy will call radiologist and say, dude, radiologist, this guy is like really important. Can you please report and tell me what's up with this patient and call emergency doctor? And the radiologist will then call the doctor and confirm, okay, this patient has left side weakness. Okay, that's correct. I see an infarct on the other side. So things like those, right? This is in general the way in which we look, and we aim to fit in this. The way we want to fit in how we use technology is, number one, help them with diagnosis, help them with AI. So this like many, so as you can see, right, there's many arrows which requires a lot of data processing. So all that will be taken care by AI, right? And then there is a lot of dissemination of alerts because stroke is a time critical disease. So there are alerts that you need to give intelligently that, hey, this patient requires your intervention. And then support and collaborate them with faster decision making, which basically gives them all the information in one place. So this is the way in which we reimagine the whole stroke workflow. Patient arrives at the spoke center, and as soon as they arrive, we know about the patient in the app, and then they'll be taken to the CT scan, and the CT scans will be available on the app, which will then be transmitted to all the people at the same time. All the different doctors at the same time, they will know what exactly they want, and then they can collaborate and decide on the treatment decisions. Right. So this is the brief for you, and I will, in interest of time, I will basically discuss how it actually works, and we'll stop there for further questions. So this is actual screenshots of our app. So how it works is, so as soon as the emergency case is done, so we will be able to take some of the information, right? What's the first name? What's the name and basic information, patient information, followed by, this is very stroke specific information. What's the blood pressure? What's the SpO2? I'm pretty sure now we heard of SpO2 now. What's the blood glucose levels? When was the patient last well-known time? And then there is this stroke scale, very specific form for stroke alone. This is called as National Institute of Health Stroke Scale. So there you basically ask questions like what's the level of consciousness, whether the patient is drowsy or alert or is in coma, right? And then you ask like, okay, how about you ask patient to close the eyes? Is he able to close the eyes? Is he able to like give you, like if you ask him current month and what's his age, will is able to answer that, right? So basic, I mean, stroke related questions. This is so this will be filled by the emergency doctor as before the CT scan is taken. And as soon as the CT scan is taken, it will be sent to our app, which you can look at the CT scan and also AI related findings and AI. So all this information, right? So this is FDA approved. AI will run the algorithm on these CT scans and will tell you what kind of abnormality is there in the brain, whether it's intraparenchymal, is there different types of stroke or different types of trauma, basically a CT interpreted, right? This is super duper accurate, 97%, I mean, plus accuracy. If you're, I think I can put up the term AUC, which is one of the accuracy metrics, right? And it's trained on like 2.2, I mean, it's trained on billion plus images, super duper accurate, probably as accurate as a resident, somebody who just got, who is not too experienced in the radiology, right? So it can detect like 12 abnormalities and it can basically create a fairly fully automated report. So this is on something called as non-contrast CT. Not only that, it can also quantify, for example, how much big a bleed is, right? And he can then also quantify, for example, if you know the brain is symmetrical. So brain left and right are symmetrical, but in this case, you see that this is not symmetrical. The left part of the brain is not same as right part of the brain. So we actually quantify the symmetry. It's called as midline shift. So we actually quantify how asymmetric. So this something called as herniation of brain. So we quantify the herniation and we can tell you like all this super quickly in your app. So then there is this specific type of stroke called as ischemic stroke. There is also something called as aspect score, which can help you identify early signs of stroke, right? Even before it can be seen on CT. So it can eliminate, basically this eliminates a subjective, this aspect score is fairly subjective in terms of humans, but AI will eliminate the subjectivity that is involved because it is dumb and it will give you same result for the same patient, right? So this is the aspect score map. I mean, I will not go into exact details of what this aspect score is, but this allows you to take the decision on whether, okay, this patient is ischemic stroke patient. And then, okay, this patient has aspect score this much. So now should I give this patient a medical treatment or a surgical treatment, right? And this is very useful in case you only have limited information. But you can also get more information called as angiogram. So what you do in angiogram is you inject so-called contrast. So if you inject the contrast, you can basically see the vessels, right? You can see the vessels of the brain. You can see, for example, like all the, white stuff, right? That's basically blood vessels. You can see that this is again asymmetrical. On the right side of the image, you are able to see the vessels, but on the left, this same vessel is missing. So this is something called as large vessel occlusion. So a significant part of the brain is not getting blood. And this, you can treat this with surgical kind of treatment called as thrombectomy. So this image is generated and automatically we can say whether this is a large vessel occlusion or not using AI. So this MIP, what you see, this is called as maximum intensity projection. And it requires some AI stuff. I will not go deep, but this can also be created using AI, and we basically detect whether this large vessel occlusion is present or not, right? And then finally, most interesting, so not only do you want to know if you want to treat a stroke patient, whether this is a stroke, whether this is an ischemic stroke or not, whether there's a large vessel occlusion or not. You also want to know if you do surgery or intervention, whether the patient will actually recover. So for this, you do this. I hope you have. I'm pretty sure you guys have heard about this fMRI thing. Similar to fMRI, there is this functional imaging called as CT perfusion. So what this CT perfusion does is it tracks flow of blood using contrast in your brain, right? It tracks your contrast, it tracks your blood flow, and it will create basically something called as Tmax and CBF. Fairly technical terms. What these do is, for example, this area Tmax, right? This shows which part of the, so this is, by the way, this is all automatically generated. This does not, you don't see them from this is all AI generated outputs. So what you see here is this part of the brain is dead. So this part of the brain, like even if you do any treatment, you are not going to do anything with it. But here, if you see this far larger part of the brain, which is less bright, right? So this is basically salvageable area. This is not getting blood right now, but if you treat it, it will get blood. All these areas that are around, right here. So this is the core basically where the blood is completely, where the brain is completely dead. So basically you can see them here. So these all you can recover, all these areas, if you treat them patient well. So this is called as perfusion imaging. And this is, by the way, it's a fun project, a course project probably, to create basic perfusion imaging for fun. So all this is we do using AI, right? So and then all this data is shown in one place because like if you remember the process, it's important. It's all of these are bit and parts of the data. The physician does not look at one thing and make decision. He looks at many different things and make decision, and all of these things are located in one single place, right? And you can actually, not only this, you can manage patients remotely using so-called hub and spoke model, even though the patient is because it's a telestroke. So because it's a stroke, you can treat them at home, but you can treat the patient at a nearby primary health or secondary health center. But the doctor who is treating them, the super specialist can sit in the, if they have access to all this data and basically do a video call, he will be able to treat a stroke, really complicated disease, inpatient complicated disease directly from a high-end center in Delhi. And this is literally what's happening. I'll skip through all this because I don't have too much time. I'll just discuss one particular, the way in which this is being used. So this is close home in Tejpur. I can also emphasize some of the US or UK, but I want to emphasize more Indian because in Indian story. So this is close home, a city called as Tejpur. This is in Assam. So this is hospital called as Baptist Christian Hospital. This is a 130-bed secondary care hospital in Assam. Right here, there's a high prevalence of hypertension, therefore huge amount of stroke prevalence. In fact, there is a specific type of complicated type of stroke called a hemorrhagic stroke, much harder to treat. This is actually 50% of all the stroke cases here. So huge burden and a lot of challenges here. No specialist. This hospital has no radiologist, no neurologist. All the stroke patients have to be treated by a local general physician, right? Non-specialist, right? What happens now with introduction of our technology is that as soon as the patient is here, they get a CT scan, and the CT scan is transferred to both the physician at the center. The CT scans report is also sent to him. All this information is also sent to Dr. Jairaj Pandian, who is sitting in Ludhiana in Punjab, right, on the completely other side of country, and he basically coordinates the treatment and tells them, hey, this patient is eligible for thrombolysis, please do thrombolysis. And this is happening right now as we speak, right, live. Patients are being impacted. Yeah, so I'll quickly, I think, yes, I have matched my time. I will not go deep, but I'm open to questions. I'm open to discuss details of anything that you are interested to know.
Thank you, Shashank. Very interesting and very real work that is affecting people's lives. We have a couple of questions here. Aniruddha, do you want to ask your question? So his question is, what AUC is required for FDA approval? Does it differ in AI for computational pathology, and is it different in India?
So number one, so I have spent about 1-2 years spending my life running clinical trials for FDA and all the other papers, clinical work I did. You need to understand that FDAs are also very sophisticated with respect to understanding AI stuff. They will reject if you say AUC is my endpoint of your clinical trial. In fact, they will not be happy if it's AUC. So they will be looking at sensitivity and specificity. So if you know the debate in the AI world, it's about generalization. The generalization is a huge difficulty right now. Even if the EUC are similar, the threshold that you use to threshold saying yes or no, and the related sensitivity and specificity, they change even if the AUC number is same across different data sets. So FDA is very specific. They are very sophisticated in this respect, and they will ask you questions a lot deeper than that. So they will be asking you sensitivity, specificity. They'll be asking you sensitivity and specificity for each abnormality that you're talking about, stratified by each kind of machine that you are targeting, the patient demographics, whether there is, I can show you a, you know what, just give me a second. I will show you one of the clinical evaluation reports that we create. I think you'll get an idea.
So just building on this question, when you say every pathology is looked at differently by FDA. So let's say if it was not a stroke, in the MRI they found, in the CT scan they found AVM or any other arteriovenous malformation.
Yes.
So would that be picked up by the AI? Would that be having the same sensitivity and specificity, or would that differ by using the same AI? So it would be like a coincidental finding, but the patient is presenting with all the symptoms of a real hemorrhagic stroke.
So they will want you to make sure that you have these confounders in your analysis. They will want you to make sure that you are calculating sensitivity and specificity on them and show that it is reasonably good, and you don't have too many false
positives. So what is reasonably good actually?
So the reasonable is the way they define it is way we have defined our endpoint, right? Is that sensitivity and specificity, its confidence interval, lower bound of the confidence interval is greater than 0.8, right? So 0.8, yeah, 0.8. So that's the, so if you are getting, I hope you understand this, all the stat stuff. So basically that's the kind of thing that they want. So it's statistically significant extremely high sensitivity and specificity. So if it's the sensitivity of lower confidence interval of 0.8, it will require you to have sensitivity and specificity of 0.95 something, because you're also limited by sample size calculations, because you cannot run trial with 10,000 patients. You will want to run trial with like reasonably 1,000 patients. You will have 100 abnormals, and then you calculate the confidence. Yeah.
So does the FDA mention this in their SOPs or guidelines, or they keep on changing, or?
Yeah, unfortunate part is, or fortunate part is, they are agile. These regulators are agile. They say, I mean, because about 3 years back, they were also quite new with respect to AI. So they did not know, for example, then that performance might differ between models of the CT scanner. They were not that savvy. Now they do. Now, if you submit an AI, I think they were not checking, for example, give me stratification by number of slices of CT scanner. Now they do. And then they'll ask you to quantify sensitivity and specificity stratified by manufacturer and how recent it is. So they're also keeping them updated. So you'll have to be on the toes, I suppose.
Okay. So this is US FDA. Indian perspective will be different.
So the thing is, in India, there is, I think, at this point of time, the regulatory scenario is not super strong. So it's treated on par with medical devices and nothing more. What I mean by that is, think that the regulatory environment in India treats AI not very different from a CT scanner or a syringe or like a injector.
But when you take an example of Tezpur in Assam as compared to any city in US, would the same guidelines apply?
So far as we I mean, abide by the highest guidelines, because we are, so far as it's like the max of all.
Okay.
So if so we'll have to work with the like most regulated environment, because
The most stringent, and they will apply it to the whole of the world. And one more thing, at the end of the report, do you mention that this has been diagnosed by Cure AI or something like as a companion diagnostic tool?
Yes, it does.
Is that required in all the reports?
It's
and
not
is that
required, but we do it for the sake of sanity, because we don't want to mislead the docs either. Because
no, understood. But do the patient have any questions about it? Is the diagnosis being made by a teleradiologist, and has Cure played a role in that? Does that affect the doctor's perception in Delhi or any other secondary look by the doctor?
So initially, there's always some, I think I will answer a bit broader than what you asked. So initially, there's always resistance. So like there'll always be some false positives and false negatives, and you want to make sure it's well communicated in terms of disclaimers that, hey, look, this requires a radiologist to double check before you make sure it's before you take complete action on the patient if you're not very confident, right? So that much we have to do disclaimer. But the way in which it's going on right now, from my from the customers I speak to, they say that earlier, I mean, I used to get patient in the night, and they used to take a CT scan of it, CT scan of the patient, and then I used to get called up in the night at 3 o'clock in the night, and hey, dude, what's up with this CT scan? Is the patient fine? Right now, what's happening right now is, as soon as we are giving AI's report to the patient, and then patient is taking to the physician who ordered the CT scan, right? Actual, like in that, you remember the graph, this emergency physician. Emergency physician is not calling anymore the doc, the radiologist. So it's yeah, it's the acceptance is, I think they're very appreciative once they get used to it, that I mean, I literally can save patient life in this. Yeah, I hope I answered.
Okay, yeah, I got it. You were showing me something on the screen. You were sharing something.
No, I'm not.
Yeah, we are already exceeding time. Let's take one more question from Professor Ganesh.
No, before that,
thanks, Shashank.
I want to say hi to you for teaching me convex optimization.
Hi. Oh,
I and all those
nice,
EM
nice.
and all that stuff, expectation maximization, I literally use that.
Okay, great. Nice to connect back to you, Shashank. Nice talk. I mean, see, when we deal with more than two classes, like N-way classification, let's say different grades of cancer, or even with stroke, I'm sure you have grades, right? Often the tendency is to ignore the, you often have a heavy tail, right? As such, most of these are finding needles in haystack, and then you can have thicker and thinner needles there. That's where I think the whole, I mean, I was happy to hear you mentioned the word agility, right? There has been agility in the metric space, but yet there is this to and fro kind of a negotiation or bargaining between what's possible, what's feasible in the AI world, and what's the user interest. Have you tried creating some kind of dashboards internally or some, I'm not talking internally in the sense of the AI developers, but I'm talking about the stakeholders, right? Where they can really find, like we talk about Pareto optimality, but maybe there are other notions of trade-off, other kind of balance points that balancing between true positives, false positives, between macro versus micro measures. So what's your experience in that space, especially with heavy-tailed distributions?
Yeah, I can. So I can completely appreciate where you're coming from, because the kind of findings you will see in an X-ray are immense. You can see up to 200 odd findings in an X-ray, and we have about 5 million, 10 million images, right? And there are some findings, 5 million, yeah, 10 million at least images of X-rays of chest. There are some findings of which we only have 200 of those in our database. So it's that 200 out of 5 million, right? So probably like 3 parts. I mean, more like 200 by 50, 10 is like 20 parts per million. So huge long tail in this space, right? So the way we work around this is that our models basically only pick the most common ones, realistically speaking. And that's the reason why this cannot be on its own. Like in the end, it's a tool. So I mean, you cannot expect COVID test, RT-PCR test to work and pick up a TB scan. I mean, TB patient. It's a tool. I mean, it's like you have a hammer and you use it to hit a nail. You don't try to eat pizza with it, for example. So that's the reason, in fact, like why we have such a disease focused way. In which, for example, I have presented stroke, right? The stroke is a focus because I have clear boundaries of what can be seen, what cannot be seen, what are stroke mimics. For example, somebody picked up AVM. That's a good example. So I know that AVM is a edge case that I will come up with my stroke patients. So we're dealing with that right now with data augmentation, finding more data, and for example, boosting and all those things. So now coming to your question of other ways of optimality, right now, as of today, the knowledge for the doctors, I mean, forget doctors, to be honest, even for most engineers in this space, if you talk to any IT guy in a hospital, if you say AUC, their mind will be blown. I mean, they are like, they were not there yet, and I don't think even explaining sensitivity and specificity is not very easy to a lot of people. So I don't expect, so we have earlier used to basically give a score to people so that they can ostensibly, they can make better judgments, right? If I give, instead of giving a 0-1, I can give them more information between 0 and 1 a number, right? But this was actually very negatively received. They did not want that because not only do they have to think about the actual patient, they also have to think what AI thinks it says, and it's extra piece of burden in their mind. So from the human interaction point of view, they prefer less thinking. I mean, so in the end, the whole idea of AI is to distill the information in the, take complicated set of information, distill it into actionable, simple stuff. That's what you have. It's a dimensionality reduction in the end. So you want to decrease dimensions as much as possible. So that's the general. I hope I have answered your question, although better around the bush a bit.
Yeah, I like that perspective of dimensionality reduction. That helps a lot. Yeah. By the way, Shashank, please do visit KCDH once. Yeah, very nice to hear from you.
I have visited quite. I mean, I come and collab with Professor Sethi quite a bit. We have actually a project.
Oh, great. Great. Thanks.
Great. Let's see. Is there one more question? Do the same principles apply to pathology too? Yes, the same principles will apply to pathology as well. Yes, Anirudh. Yeah. There, the added thing is that you have a very large image, and then it's also a spatial needle in a haystack. So that is the reason that, and then it is not natively digital. People are so used to looking at glass slides and not investing in digital scanners that it has lagged. AI in pathology has lagged that in radiology quite a bit, but I think it is soon picking up. Okay, with this, let's conclude this session. I want to thank both Nipun and Shashank for very great and informative talks, and to all the participants for asking these questions to make it lively. So with this, we are concluding this session. Thanks, everyone.
Yeah, I'm also thankful to the Professor Amit, who is chairing this session very wonderful, in a very wonderful manner, and drive the session very smoothly. Thank you, Professor Amit.